BXD family. So with that, I will hand it over to David, and I will give you a heads up around 12 or 13 minutes just to let you know that you're nearing the end of the time.
Great, thank you very much for that introduction, and thank you all for thank you for the committee for inviting me to talk, and thank you all for coming to listening. So I'm going to talk to you today about gene by environment by age effects with mitochondrial DNA copy number, aging and longevity in the BXD family. So why are we interested in aging? Well, the human population is getting ever older. The worldwide average lifespan is now about 75 years. We've doubled the number of people reaching 95. And by 2050, we expect that over 3.5 million people will reach over the age of 100. And there is a genetic contribution to longevity. So the heritability is about 20 to 30%. But it's not really aging that's important, it's healthy aging. I want to get to 100 being physically and cognitively fit. And mitochondria are really important for aging. So there's the mitochondrial theory of aging, but everything from cell cycle, oxidative phosphorylation, oxidative stress, these are all involved in mitochondria. And every cell has many copies of mitochondria, to just 2 copies of the nuclear genome, and different tissues have different numbers of mitochondria. And that's related to their metabolic needs, so that's especially high in the CNS. Neurons can be packed full of mitochondria because they need that energy. And mitochondrial DNA copy number has been linked to many diseases of aging. So particularly interesting for this audience is neurodegenerative diseases. And we know that mitochondrial DNA copy number changes over time. So it sort of increases slowly during adulthood and then drops off rapidly in old age. And this has been linked to both healthy physical and cognitive aging. So if you have a higher mitochondrial DNA copy number, you tend to have healthier aging and better cognitive function in old age. And it's also been related to Alzheimer's disease. So patients with Alzheimer's tend to have lower mitochondrial DNA copy number than their healthy counterparts. So there's many things that influence longevity and mitochondrial DNA copy number, and isogenic strains are a great way to study all of these things. So obviously we can do genetics, we can have related individuals and many replicates within the same genome to map modifiers. We can look at diet and other environmental effects, because we can have basically an infinite number of replicates of the same genome on different environments. We can look at age, because we can pull out a brain at 6 months or at 24 months in a genetically and environmentally identical individual, which you can't obviously do if you've only got the one individual. We can look at sex, because in these isogenic strains, basically everything is identical bar the sex. It's only really the non-pseudoautosomal part of the Y chromosome that differs. So we can look at every combination of genome, environment, age, to get this whole picture. And the way we've done this is using this aging BXD cohort. So the BXD, as many of you all know, is a cross between the C57 Black 6 and DBA2J, which is recombination. These are then inbred for at least 20 generations. So we have between these 140 and 150 lines. Every individual within the line is genetically identical, and each of these lines is a unique mosaic of B6 and D2 alleles. So we took 87 of these BXD strains and both their parents, majority of which were female, and put them either on a standard chow diet or a high-fat diet. And then about 2/3 of the animals we allowed just to live out their life. We just saw how old they got, and we could get a genetic measure of longevity. How old does a strain live just naturally? And for about 1/3 of these, we then sacrificed them at about 6, 12, 18, or 24 months of age and stored a broad range of tissues. But for this study, I'm going to sort of concentrate on the liver. So this is obviously a huge cohort, really interesting and being studied from many angles. So Sahita has a paper in review looking at longevity. Jose and Benny have a paper on the methylome and the epigenetic clock in this population. Evan is looking at a transcriptome and a proteome and a metabolome. And obviously you can bring this all together to look at QTL mapping and systems genetics. I'm interested in this sort of mitochondrial DNA copy number angle. So how do we look at this? Very easy. We took 3 primers in different positions around the mitochondrial genome, 2 primers in the nuclear genome, used quantitative PCR to get an estimated number of copies of those, and then looked at the ratio of mitochondrial copies to these 2 nuclear copies. This is preliminary data. It's still data that's coming in. So far we've done 301 females of this 662 total individuals, and we get a good heritability, 0.36. As you can see, about 2/3 of what we've done so far is chow diet, only about 1/3 is high fat, and we've not done any strain completely. So we kind of have a patchwork of strains and ages and diets. And when you plot that out with estimated mitochondrial DNA copy number and age at death, you kind of don't see a lot. You can see the approximate ages we collected, but if this was just a population of individuals that you didn't know anything about their genome, you'd say not a lot is happening. But because we know these strains, we can actually see that there's a really strong and significant strain effect, a really strong and significant strain by age effect, and a suggestive diet effect, which we'll see when we balance out the diet and what happens with that. So what does this sort of strain by age effect mean? So if we imagine that this is a trajectory of one strain of their mitochondrial copy number of their age, you have another strain that does something like that, another strain that does something like that, and another strain that does something like that. So now if you look at the whole thing, you can't say one strain is higher than all the others, but any particular age, you could say that this strain is high, this strain's low, whereas over here, this strain's high and that strain's low. So that's that strain by age effect. So when we cut things down to just look at particular age ranges, so for this approximately 18-month range, our heritability jumps up hugely. We get to 0.82, and you can see that these biological replicates cluster really well together, and they are different from each other. So you can see this strain by age effect. And that high heritability is maintained for all the different combinations of approximately 6, 12, 18, or 24 months of age on the chow diet and high fat diet. And we can now do our QTL mapping. And we find 2 significant QTL, one for mitochondrial DNA copy number on the high fat at 6 months and one at 18 months. So when we look at that, it reaches significance. And you can see that those strains that have the maternal allele in red here have a low value, and those that have the paternal allele in green here have a high value. So this is only with 19 strains. At the 18 months, we're only at 16 strains, but again, you can see the same pattern at this region on chromosome 11. Those are the low value, have the maternal allele, those with the high value paternal allele. And we're probably going to double or more the number of strains. So hopefully we'll get some more recombination in this area to narrow down the number of genes within this loci. But also we have the whole stack of omics. So we've got the genome, the epigenome, the transcriptome, the proteome. So we can narrow down really well to candidate genes. However, with this preliminary data, what's been really interesting so far is a significant correlation between mitochondrial DNA copy number at 18 months of age and longevity, both on chow diet. So you can see that here. And the mean longevity across strains is about 680, so somewhere here. Whereas when we're measuring this mitochondrial DNA copy number, it's somewhere between sort of 480 and 600 days. So we can imagine that these strains down here are somewhere on this end of the their trajectory through life. They're living longer than they should be genetically, so their mitochondrial DNA copy number is low. These in the middle are somewhere on this part of the trajectory, and then these at the top are still around their peak, so they've still got plenty of time that they're going to keep living out. So I think this is really interesting, the fact that we have a biomarker of longevity. And because we have this BXD cohort, it's hugely expandable. And the first place I want to expand it to is tails. which may not seem very interesting tissue to most of you, but all human studies use a peripheral tissue to try and find out something about the central tissue. So they take blood or buccal epithelial cells, whereas in this, we can actually check that. We can see if variants that affect mitochondrial DNA copy number in the liver or brain also affect it in the tail, or different environments or different ages. And if this tail works, it means that this is hugely expandable, because different projects take tails, clips for genotyping, which means they already have DNA to do this kind of analysis. And the first place we want to take this to is the AD-BXD model. So this crosses an Alzheimer's disease model, the 5X FAD, to the BXD to produce a variety of genetically diverse strains and their non-transgenic controls. So these replicate many features of Alzheimer's disease, both physical and cognitive features, and you can read all about them here. But what's cool for me is they have all sorts of tissues stored, but they also already have the tail clips for genotyping. So they have 3 time points, entry in the study, and then when they were culled either 6 or 14 months, and have males and females. This means that we can rapidly expand to look for sex effects, sex by strain, sex by environment, sex by age, because we know that sort of women live longer than men. We have additional samples for our QTL mapping, and we can also look for individual correlations now. So if you have low mitochondrial DNA copy number at young age, does that predict what's going to happen in older age? We can see differences between the non-transgenic and the AD-BXD. So in Alzheimer's disease patients, we know they have lower mitochondrial DNA copy number. Is that reflected in this model? And is there correlations in normal phenotypes? So the non-transgenics over age were measured for all sorts of learning, memory, and cognitive phenotypes, and we can see if mirrors that healthy or unhealthy aging in humans. Another cool place we can take this to is using the intervention testing program, ITP, Het3 animals. So this is basically an F2 created from 3 parents, and they tested all sorts of interventions that are related to aging. So diet, rapamycin, other drugs. And what's good for us is the D6 and the D2 are 2 of the parents of that population. So any QTLs for mitochondrial DNA copy number, longevity, found in the BXD should replicate in this Het3 population. And it also adds in that diet and drug as an environmental effect. So we can look for the G by E. So conclusions, we've seen really strong strain and strain by age effects on mitochondrial DNA copy number. Mitochondrial DNA copy number correlates with the longevity in the BXD. And we have this huge stack of omics data in this population that we can start to use to look for gene by environment, by age interactions. And this is an expandable system. It's not just one thing that's done and gone. It can be expanded into other places. So my time's getting close, so I'm going to thank everyone involved in this project, especially Jessie, who's done the lab work, and Casey and Melinda, who've looked after the animals, which you can imagine is a huge project. And I will thank all of you for your attention. I'm happy to take any questions now or in the future.
Okay, thank you, David. That was an excellent presentation. Do we have any questions? If you want to just unmute yourself. Anyone?
I have a question. So
Sure.
when you're doing this type of, in cross between the 2 strains, did you look at the sort of, I guess sort of the variation in the phenotypes in the parental strains? And obviously there's probably modifiers, masking genes that would come out of it, but is there a sense of, okay, when there's this large strain difference ahead of time, we're going to get more QTLs or more information?
So I don't know. We obviously have the non-transgenic controls with this crosses, or do you mean in the BXD in general?
Well, for this particular one, I don't know if you showed the parental strain data. I'm guessing you might have it. And then just in general with this type of these type of crosses, are you going to have better outcomes if there's a large strain difference between the parents, or is there enough sort of modifiers that when you start shuffling the deck, you get stuff coming out all the time?
Yeah, exactly that. That even if there's not much change in the parents, you can still have variation in the population. So I'm not sure if you can see this at the top of the screen now.
Yeah,
I think I have a
on the left side there.
Yeah. So this is A beta in the BXDs. So you can see that the two parents are basically indistinguishable, but there's this huge variation in the BXDs. So although it's a nice sign if there's difference between the parents, it's really not necessary because there can be enough variation sort of within there.
Thank you.
Okay, thank you, David. I think we can move on now to our next speaker. And our next speaker will be Christiane Gaines, and she is from the School of Medicine and Department of Genetics at the University of North Carolina in Chapel Hill from the Tarantino Lab. And the title of her talk is Assessing the Impact of Compositionally Distinct Gut Microbiotas on Differences in Initial Cocaine Sensitivity in Closely Related Inbred Mouse Substrains. So Christiane, if you want to take it away.
All right, can everyone see my screen okay?
Yeah.
Yeah, okay. All right, thank you so much for the introduction. It's nice I can skip this slide. So today I'm really excited to share some of my thesis work. And so substance use disorders can be defined as the recurrent use of drugs causing clinically and functionally significant impairments. A recent report from SAMHSA in 2018 found that there were 1.9 million cocaine users aged 12 and above in the past month. When we look in the literature, we see that both genetic and environmental factors can influence your SUD risk. And so on the genetic side, we see that heritability estimates from SUDs from twin studies can range from 0.4 to 0.7, with cocaine having the highest heritability estimate at 0.7. There are also environmental factors that can influence your SUD risk, and these include maternal care, family environment, as well as the gut microbiome. And so I'm interested in initial sensitivity to psychostimulants, as this is shown to predict future use. So we see that individuals vary in their initial response to drugs of abuse. Retrospective studies have also shown that persons that have greater positive initial drug responses, this can predict their latency to subsequent use as well as abuse. And so I'm really interested in investigating the role of both genetic and environmental factors in order to better understand initial cocaine sensitivity. And cocaine is a behavioral stimulant. So in order to model initial cocaine sensitivity, we can do that looking at cocaine-induced locomotor activity, which can be measured in the open field. And so the open field is a 4-walled plexiglass chamber that has infrared beams. And so once you place a mouse into this chamber and it moves, it makes breaks in these beams, which generates these traces and allows us to measure the distance moved of that animal for a specific period in time. And so to measure this, our lab uses a 3-day acute sensitivity protocol. And the first 2 days, the animals receive intraperitoneal or IP injections of saline, and on day 3, they receive a IP injection of 20 mg/kg of cocaine. And then at the end of each test, into the open field for 30 minutes to measure their locomotor activity. And so in my project, I'm using inbred mouse substrains, and substrains are mice that have been separated and maintained separately for more than 20 generations. So here I'm showing the C3H progenitor along with 2 of its substrains, the C3H-HEJ substrain and the C3H-HE-NTAC substrain. And so substrains differ only for polymorphisms that have existed prior to the strain's divergence or accumulated since as a result of random mutations, and this can give rise to different phenotypes. And so traditionally, mapping studies would use inbred mice that were phenotypically as well as genetically divergent. And one of the challenges with this is that you can have hundreds of genes or hundreds of thousands of potentially causal SNPs, which makes it really difficult to narrow down causal SNPs. And so by using substrains, because they are closely related, they provide a more simplified system for identifying and validating functional variants, as well as their impact on complex traits. And so this leads into the aims for my project, which were to first identify substrain differences for acute cocaine-induced locomotor activity. Then I wanted to identify genetic polymorphisms as well as environmental factors that may be responsible for locomotor activity differences that we may observe between these substrains. And so using the two substrains I explained earlier, we tested a cohort of these mice using that 3-day acute sensitivity protocol. And we see that on the first 2 days, the HE NTAC substrain had significantly higher locomotor activity in comparison to the HEJ substrain. On day 3, once the animals receive an acute dose of the drug, we see that the HE NTAC substrain had significantly higher cocaine activation in comparison to the HEJ substrain. And so this finding was actually replicated across multiple cohorts of these substrains. And so next we wanted to see if we could identify polymorphisms that were responsible for this difference that we had identified. And so to do this, we used our substrains to generate an F1 population, and then these mice were further crossed to produce a F2 mapping population. And so using mice from each of these populations, we phenotyped these mice using that 3-day acute sensitivity protocol. We genotyped our F2 mice using the Minimuga novel genotyping platform, and then we performed QTL mapping using our QTL. And so here I'm showing the phenotypic data for day 3 once the animals receive the drug. And we see that the F1 and F2 populations exhibited cocaine-induced locomotor activity similar to the HEJ parent. So this showed us that having low cocaine-induced locomotor activity was dominant. And so we continued on to do QTL mapping. And here I'm showing day 3, again, once the animals received the drug. And we had genotyped 387 animals, and we only were able to identify a suggestive QTL on chromosome 19. Now, when we look at the effect plot for this QTL, we see the direction of the effect plot matches that we observed in the parental substrains, where mice that are homozygous for the NTAC alleles have a higher locomotor response in comparison to those that are homozygous for the HEJ alleles. And so because we had genotyped this many animals and identified the suggestive locus, maybe we thought that there was some environmental factors that were leading to this behavioral difference, or maybe there was there's an environment by genetic interaction that might be playing a role. And so lastly, this led us to trying, like I mentioned, to look at environmental factors. Now, when we look in the literature, we saw that the gut microbiome and maternal care can influence cocaine sensitivity and reward. And so we wanted to look specifically at these two factors. And so there was this really interesting study in 2016 that found that if you took B6J mice and gave them a cocktail of antibiotics, this can lead to an overreduction in their gut microbiota. And this can increase the rewarding properties of cocaine as well as behavioral sensitization. When we look at maternal care, this can actually influence behavior in adult offspring, and it can also influence cocaine-induced locomotor response. And so I was then interested in looking at the composition of the gut microbiota between these two C3H substrains to identify any role that it may play in the activity differences that we observed. And so to do this, I collected fecal boli from both substrains. So for the HEJ substrain, I collected samples from 10 females, 21 males across 12 different cages. And then for the NTAC substrain, I collected from 14 females and 14 males across 9 different cages. And so I extracted DNA, we performed 16S sequencing, we QC'd the samples, and we looked at the abundance of microbes for each sample, as well as we characterized the diversity of the gut microbes. And so here I'm showing a principal coordinate analysis with Bray-Curtis dissimilarity. And so each sample, the color represents the substrain that sample came from. Squares represent female mice and circles represent male mice. And so the way you would read this graph is that samples that are similar would cluster closer together, whereas samples that are less similar will cluster further away from each other. And what you can appreciate here is that all of the samples that came from HEJ mice clustered together as well as separately from the HE NTAC substrain. And so just to note, these mice were caged by substrain with the same environmental conditions. And so they were in the same room, they were bred in the same room, they were handled by the same individual, they were also given the same diet. And we see that the community of microbes was unique between these substrains. We were also interested to know whether samples that came from these mice, if there was any clustering based on the cage that these samples came from or sex, but that was not observed. And so this is really showing us that the difference in the microbial community is driven primarily by substrain. And so because these animals were caged by substrain, we were interested to know whether if we co-house these animals, would that adjust the microbial community? And so to test that, we co-housed these animals at weaning. And so in a single cage, we had both substrains being represented. And then once these animals reached adulthood, we tested them using that 3-day acute sensitivity protocol. And we still saw that difference following cocaine. And so next, we assessed the gut microbiota of these mice, and we saw that the community of microbes, again, was still unique between the substrains, where the HEJ substrains, they clustered separately from the NTAC substrains. And so we were interested to know whether this, the unique clustering of these microbes was because these mice were co-housed at weaning, and maybe it's too late in order to shift the microbiome. So maybe if we actually co-house these animals when they were younger, this can actually induce some shift. And so to test this, what I'm currently in the process of is performing cross-fostering experiments. And so cross-fostering in literature has been found to induce a permanent shift in the offspring microbiota to match that of the nursing mom. And so these are the 4 different cage types that will be represented in this experiment, where we will have an HEJ mom that has HEJ pups from another HEJ mom, or we can have an HEJ mom that has HEJ pups from another HEJ dam, as well as NTAC pups. And so in this experiment, I will be collecting fecal boli from both the dams and the pups. And then when the pups reach to adulthood, I'll be testing them using that 3-day acute sensitivity protocol. And so by having the behavioral data and then us assessing the gut microbiome, we can see whether it plays any role in the cocaine-induced locomotor activity differences. Now, in parallel to this, I'm also performing maternal care monitoring of these cross-fostered cages. And so from PND 2 through 5, I am visually assessing these cages twice a day for maternal care behaviors, which include nurturing, non-nurturing, as well as self-maintenance behaviors. And so just in summary, I was able to identify this really striking difference between the substrains for cocaine-induced locomotor activity. We were interested to know how environmental factors such as the microbiome, maternal care, and genetics might play a role in the behavior that we observed. And so far, it was really surprising that we had identified this suggestive locus on chromosome 19. This really interesting study, we found that the microbiota was unique between these substrains. And currently we're in the process of performing cross-fostering as well as maternal care experiments to see essentially if by cross-fostering these mice, you can have a shift in the microbiome and how this would in fact affect the behavior of these mice in adulthood. And so this was a pretty big undertaking, and I just want to make sure I take time to acknowledge all the people that was involved in on all of these experiments. The Tarantino Lab, the Ferris Lab, Fernando Lab, Carol Lab, my committee members. Thank you to the IBANGS Trainee Planning Committee for allowing me to do this talk today, as well as our funding sources. And with that, I'll be happy to take any questions.
Okay, looks like we have some questions in the group chat. The first question, I believe Carl's question was answered during the talk. So from David Ashbrook, he says, very cool. Are there known maternal care differences between the parents?
Yeah, so I have tested a small group of these animals so far. And so far, doesn't really seem that there are any major maternal care differences, but again, this was across only like 6 cages. I'm in the process of testing a lot more, actually doing that today. So hopefully I'll have a lot more data on that very soon. All right, so okay, I can look at the next question.
Oh, yeah.
Did you check for, okay, so the question is, did I check for food consumption? Could that explain the difference you see? We did not check for food consumption at all. The animals had like unlimited access to food at all points in time during the experiments. I'm actually not sure if that could explain the difference that I see between the substrains, but it's something that I can look into.
There's one more question here that's coming up. We're running a little short on time, but if you want to quickly answer one of these questions.
Okay, I know you mentioned that there were no sex effects, but I was wondering if there was a sex by genotype interaction. It looked in your PCA that there might be an effect in C3H strain. So for the PCA, okay, so the principal coordinates analysis, I have not looked to see if there was a sex by genotype interaction. I mean, that's something I could look at. At least for the phenotypic data, we have not seen any sex by genotype interaction. But yeah, that's something I can go back and look at.
Okay, thank you, Christiane. Now I'm going to hand it over to Danila, who will introduce the next two speakers.
Hi, everyone. Thanks for being here. So our next speaker is Dr. Sam-Moon Kim from the Center for Systems Neurogenetics of Addiction at the Jackson Laboratory in Bar Harbor. And he will be talking about the high throughput phenotyping reveals possible relationship between circadian rhythms and cocaine addiction in genetically diverse mouse populations. So with that, I hand him over.
Can you hear me? Okay.
Yes.
First of all, thanks organizer for giving me an opportunity to share my data at this meeting. So I'm gonna start with a brief background about the circadian rhythm. So because of rotating Earth, all living organism on the Earth has the rhythmicity in their physiological process and their behavior, which is called the circadian rhythm. So this circadian rhythm is regulated by like a cell autonomous molecular clock. So this cell autonomous molecular clock is composed of positive and negative, like transcriptional and translational feedback loop. And this one cycle of the transcriptional and translational like feedback loop takes about 24 hours. So that's why we have the circadian rhythm period about 24 hours. So among these all different components, BML1 has very important role to maintain the circadian rhythmicity in our body. So for example, so these are the ectogram of wheel running activity in wild type mice and BML1 knockout mice. And this white background indicates the light on, and this gray background indicates the light off, and this black dot indicates the mouse is running on a wheel. So as we expected, since this mouse is a nocturnal animal, so this onset of the mouse activity is well entrained to the light off in the light and dark cycle. But under constant darkness, their activity onset started earlier and earlier compared to the previous day because mouse has, in general, mouse has a slightly shorter than 24-hour circadian period. But however, so when we look at the BML1 knockout mouse, we can see that a rhythmicity in the behavior indicating BML1 is very important to maintain circadian rhythmicity in the mouse. So therefore, our study measured this BML1 transcriptional activity as indicator of a circadian clock. This circadian clock regulates almost every physiological process, including drug addiction. But this regulation is not just a one-directional regulation. This drug abuse can feedback on the circadian clock and like the disrupt the circadian clock mediated the timekeeping mechanism. So disruption of circadian clock exacerbate like drug abuse. So it kind of contributes to the drug addiction. So therefore, it's important to understand the mutual interaction between circadian clock mechanism and drug addiction. So to understand the genetic mechanism underlying these two mutual interactions, we use the powerful animal model, which is the collaborative cross and diversity outbred mouse. This CC and DO mouse population started from 8 founder strain and 5 common inbred strain and 3 wild-derived strain. And our center focused on the phenotyping cocaine addiction-related behavior in this mouse model, but our lab specifically focused on the phenotyping of circadian rhythmicity in these mice. But our initial question was, how can we measure the circadian rhythm in these many mice? So in 2005, US Sibler's lab published one really interesting paper comparing 2 different circadian rhythm in the same mouse, the behavioral rhythm and the fibroblast rhythm. And they found a high correlation between these 2 different rhythms in the same mouse. For example, when the mouse has longer circadian period in the behavior, they also found longer circadian period in the fibroblast rhythm and vice versa. So this paper suggests that fibroblast is really good model for the screening circadian rhythm. So we adapted this idea to do the high throughput cell-based screening of circadian rhythm in our animal population. So we got the ear biopsy from each mouse, and then we isolate the fibroblast, and the fibroblast received lentivirus expressing the BMEL1 fused to the luciferase gene. So by measuring this luciferase gene by these luminometers, we can study BMEL1 transcriptional activity. So this one is an example of a fibroblast rhythm we get from this bioluminescence. And once we get this data and we analyze all different circadian parameters like the period, amplitude, and phase and damping rate. So period is the time to take one cycle of rhythm. So basically the measurement of a peak-to-peak interval, and amplitude is the how big of this rhythm. So the measurement from trough to peak, and phase is a specific timing when the rhythm has the highest amplitude. So indicating delayed rhythm or advanced rhythm, and damping rate indicate how quickly the rhythm goes down. So indicating the persistence of rhythm. So here is 2 representative fibroblast rhythm we found in the founder strain. So first, we observed strong oscillation of fibroblast rhythm in all founder strain of a mouse. And the second, when we compared this the rhythm between the biological replicate within the same strain, we found consistent rhythm trace between the biological replicate, indicating reproducibility of our data. And more importantly, we found strain differences in this molecular rhythm in the fibroblast among the founder strain. For example, AJ mouse and NOD has really low amplitude compared to 120S1 and WSB mice. So in order to understand more detail about these strain differences in fibroblast rhythm, we quantified all circadian parameter data and the plot on the bar graph. And this filled bar is data from the male mice, and this empty bar is data from female. And we compared all these data to C57 black 6J mice, which is the most common inbred strain in the lab. And from this data analysis, we found the strain by sex interaction of this fibroblast rhythm in period and the phase. For example, the castor mouse has a significantly shorter circadian period compared to these 6 mice, and AJ mouse has a significantly longer circadian period compared to these 6 mice in the male. For the female, we also found significantly shorter circadian period in castor mouse and 129S1 and WSB. And also we found advanced circadian rhythm in the castor mouse compared to B6 in male. And also most of the strain shows advanced rhythm compared to B6 mouse in the female group. Interestingly and surprisingly, we also found reproducible sex differences in the period and the phase. For example, the female has a longer circadian period in the B6 mice compared to the male, and female has a shorter circadian period and advanced rhythm in the female compared to the male for this PWK mice. In addition to the period and the phase, we also observed the strain differences in the amplitude and goodness of fit. And I forgot to mention about this goodness of fit. Goodness of fit indicate how well our observed rhythm fit to the hypothetical sine curve. So basically it indicates the robustness of the rhythm. So from this data, we calculated the heritability estimate, and we got the strongest heritability score per period, like 35%, and relatively strong heritability for amplitude and phase and goodness of a fit. These all data together indicate genetic diversity contributes to the phenotypic variability in our circadian rhythm. So at this point, we want to further confirm that the fibroblast rhythm we observed in our study is truly represent the circadian rhythm in mice in vivo. So we focus on two extreme strains, AJ mouse and cast EIJ mouse, because regardless of sex, AJ mouse always shows longest circadian period, and cast mouse always shows the shortest circadian period. With these two mice, we measure the real running behavior in light and dark cycle and constant darkness. So in AJ mouse, all the mice are well entrained. Activity onset of this AJ mouse are well entrained to the light off, but under the constant darkness, their activity occurs earlier and earlier every day, indicating less than 24-hour circadian period in the males and female. But cast mouse show little interesting phenotype in their behavior. Like you can see here, cast mouse activity onset occurs like 2 or 3 hours earlier than actual light off, indicating it's not really well entrainable like a mouse. But under the constant darkness, same as AJ mouse, the activity rhythm shows like the period of activity rhythm shows less than 24 hours. However, when we compare this AJ mouse and cast mouse, the slope of this activity onset under constant darkness is way more dramatic in cast mouse, indicating cast mouse has a significantly shorter circadian period than AJ mouse. Here is the quantified data from this ectogram. Like we observed in the fibroblast rhythm, cast mouse has a significantly shorter circadian period in male and female, and also cast mouse has a significantly higher amplitude in real running activity in male. So all this data together, like with the fibroblast rhythm and this behavior rhythm, so all this data together indicate the fibroblast rhythm we observe from the founder strain represent the true circadian rhythm in mice. So I'm gonna switch gear a little bit to the CC strain. Among the CC strain, we focused on two different CC strains, CC04 and CC41. The reason why we focused on these two CC strains is like Sarah at the UNC did cocaine locomotor sensitization, and the Price at Jackson lab did cocaine IBSA study, and they shows that this CC4 mice shows more vulnerability to cocaine addiction. So that's why we wanted to know if these mouse show also different phenotype in circadian rhythm. So first one is the molecular rhythm in the fibroblast. This black line represents the data from CC4, and this gray line represents the data from CC41. And here's a quantified data. And quantified data shows that CC41 had a significantly longer circadian period compared to CC4 mice. And also amplitude of CC41 is significantly lower in male compared to CC41 of CC4 mice. In addition to this fibroblast rhythm, we also observed the behavioral rhythm, which has the same pattern as a molecular rhythm. And quantified data showed that CC41 has longer circadian period compared to CC4. And also the amplitude of CC41 is significantly lower than CC4 in the male group. So we don't know the specific mechanism of this thing But this data somehow suggests that there is some correlations between circadian rhythm and cocaine addiction. The last thing that I want to share with you is like the DO data, the fibroblast rhythm.
You have 2 minutes left.
This one is the last slide. And we screened fibroblast rhythm from more than 300 DO mice, and then compare the distribution of a circadian parameter to the founder strain. And one sentence summary is DO mouse population display more phenotypic diversity in the molecular rhythm than the founder strain. So for example, in the period, as you can see here, it's more than 80% of a founder strain show the circadian period about 24 hours, but only 20%, 25% of DO mouse displayed like a period about 24 hours. And the range of this circadian period is way bigger in the DO mouse compared to the founder strain. And also we observe the same pattern of this high diversity in DO mouse in phage and damping rate and goodness of fit. So this more phenotypic diversity in DO mouse is possibly because of there is a heterogeneity in DO mouse genetic background. So to summarize all this data, so we observe the strain by sex interaction in our fibroblast rhythm, and we also find the strong heritability in the molecular rhythm data. And we also observe the DO mouse population display more phenotypic diversity compared to the founder strain. And these are ongoing study right now. And we are working with the Jackson Lab, like BBAG is working on trade correlations between our circadian data and addiction-related behavior, and also QTL mapping for the molecular rhythm. And also we are in the middle of a sample preparation to study the impact of genetic variation on circadian transcriptome. And I'd like to thank especially for my wonderful mentors, Colin and Ryan, and also the Price who did the IBSA, cocaine IBSA, Jackson Lab, and BBAG calculate all our heritability score and the working on correlation and QTL mapping. And Leona sent us the sample for the fibroblast isolation, and Sarah did all those like locomotor sensitization study with the cocaine. Here is my funding source. I'm happy to take any question.
Thank you. Very good talk. I think we don't have time for questions, so I'm gonna go ahead and introduce our last speaker for the session, which is Kristen Borrelli. She's a PhD candidate from the neuroscience program at Boston University. And she will be talking about neonatal morphine in CFU mice induces behavioral signs of withdrawal, sex-dependent transcriptomic profiles in brainstem, and altered affective and psychostimulant-induced locomotor phenotypes in adolescents. And with this, Kristen, you can take over.
Okay, let's get this on. Start my timer. Okay, so thank you for the introduction. Everyone can hear me, right? Yeah, okay.
Yes.
So as you all probably know, there's an ongoing opioid crisis in the United States where opioid-related overdose rates have drastically risen over the last 20 or so years. And along with this, we've also seen a drastic rise in maternal opioid use disorder and the birth of neonates that are dependent on opioids and are suffering from withdrawal symptoms when they're born and no longer exposed to the drug. So this has been termed neonatal opioid withdrawal syndrome, or NOWS. And the various symptoms include irritability, insomnia, inconsolability, and increased vocalizations in the neonates. And we don't really know in detail what the long-term effects of perinatal opioid exposure are in terms of neurodevelopment and behavior. And these things are challenging to study in a human population. However, mouse models offer a more efficient way and obviously offer far more environmental control to study these long-term effects. So we've developed a model of third trimester equivalent opioid exposure in outbred CFW mice, where we're doing injections postnatally from postnatal day 1 through 14. And again, this is roughly equivalent to the third trimester of pregnancy. And I should say that this exposure during this period is necessary for the presentation of NALS symptoms. And we're injecting the pups twice a day with 15 mg per kg of morphine sulfate, and these are subcutaneous injections, or saline as a control group. So everything is being done in a split litter design. That way we can control for the effects of maternal care a little bit better. So half the pups are getting saline in 1 litter and half of them are getting morphine. And we developed this model, and it was inspired by Julie Blendy's work at the University of Pennsylvania, and there's a recent publication that you can read more about that there. So we did several different phenotyping assays during this injection period from postnatal day 1 through 14, and then we also did some adolescent testing, as well as RNA sequencing on brainstem tissue at postnatal day 15. So all of these tests are being run during a state of spontaneous withdrawal in the pups, approximately 16 hours after the last morphine injection. So we're looking at rates of ultrasonic vocalizations as an indication of distress in the pups. We're looking at thermal hyperalgesia in the hot plate assay, as well as the tail withdrawal assay. And we're also looking at different affective phenotypes, including the open field at P21, and again, more behaviors during adolescence. So quickly to summarize our behavioral findings, we see really drastic weight deficits in our morphine-treated pups, and this lasts not only through the injection period, but also to the point of weaning at postnatal day 21 and to adulthood at postnatal day 51, early adulthood. In terms of ultrasonic vocalizations on postnatal day 7, there are no differences in the overall number of USVs, but when we break it down into minute bins, we see that our morphine-treated females are vocalizing more during the first few minutes of testing. And that's one of the interesting sex effects that we see. However, on postnatal day 14, both the males and females in the morphine-treated groups, which are these dotted lines, are vocalizing significantly more than the saline-treated controls. So after the USB testing, we did this testing for pain sensitivity, and we found at both P7 and P14, our morphine-treated animals showed reduced latency to show pain responses in the hot plate assay on P7 and P14, as well as the tail withdrawal assay. They're also showing decreased latency to withdraw their tails from a hot water bath on postnatal day 7 and 14. So this increase in pain sensitivity supports the notion that these mice are indeed in spontaneous withdrawal from opioids. And it kind of is a way for us to validate that we're actually producing what we want to produce in these mice. So on postnatal day 21, we did open field testing, and you could see that the mice are both traveling less in the open field, and they're also spending less time in the center of the open field. So this indicates, 1, that they're showing less exploratory behavior, and 2, that this could indicate if they're spending more time on the edges of the arena, that they're more anxious at that point in time. So again, we did this behavioral battery during adolescence to look at more long-term or prolonged effects of the perinatal morphine exposure. And briefly, I'll talk about some of these different assays that we're doing. I'll focus on the light-dark testing, the elevated plus maze or EPM, the Barnes maze, and this sensitization, locomotor sensitization assay that we ran. So for the Barnes maze, if you're not familiar, this is done on an elevated platform. The mice are trained to identify a target hole. So this is viewing it from above, and the target hole has an escape hatch located underneath of it. So during training, they'll run to the target hole and go into the escape hatch. Then on the probe day, you remove the escape hatch from underneath of here. And the data that I'll show is the average distance of that mouse on that probe day from the target hole. So this is a spatial memory task to see if they remember where that escape hatch was. The light-dark test, you might be familiar with this, where we're looking at the aversion of the mice to spend time in the light side of a chamber. And this elevated plasmase or EPM, again, looking at anxiety-like behavior, impulsivity, the tendency for them to spend more time on the open arms, or the outer portion of the open arms would be indicative of reduced anxiety or increased impulsivity. So briefly, the results of these tests, we see in our Barnes maze assay that our morphine-treated mice are significantly further away from the target hole on that probe day of testing. So this would indicate that they could have deficits in spatial memory that are sustained into adolescence. In the light-dark assay, while they don't show differences in the actual time that they're spending in the light, when we look at their behavioral tendencies while they're actually in the light side of the chamber, we see, and this is a heat map of activity on the light side where the dark side would be over here, we see that the saline-treated mice spend a lot of time in the threshold to the light, whereas the morphine-treated mice on average are exploring the light side of this chamber a little bit more. So on average, they're located further from the threshold to the dark side of the chamber, which could indicate that they have less anxiety or increased impulsivity-like behavior. So again, we found something similar in the EPM assay where our morphine-treated mice in light gray here are spending significantly more time in the open arms of the EPM and more time in the outer open arms of the EPM. And this is close to significant, but not quite significant in the closed arms. So again, the same implication that we saw an indication of here in the light-dark assay, that they have decreased anxiety-like behavior or increased impulsivity. So lastly, we did a locomotor assay in response to 2 mg per kg of methamphetamine, which was delivered IP. So we established baseline locomotor activity on days 1 and 2, and then for a period of 3 days and over 60 minutes, we're looking at their locomotor activity following injection with meth. So we can look at their locomotor activity relative to this baseline locomotor activity on day 2 of saline. So here this is % day 2 distance, and you can see that our morphine-treated mice in light gray have significantly greater increases in their locomotor activity relative to their saline-treated litter mates. So this could, while it doesn't speak to reward sensitivity necessarily, this could indicate underlying differences in the development of dopaminergic innervation. And this is something that we're starting to look into now. I'm working on intracranial self-stimulation studies in these adult mice that have been treated with morphine. So lastly, we did brainstem RNA sequencing in these mice. So we collected tissue at postnatal day 15. And interestingly, we see really distinct differential expression profiles between our male and female morphine-treated mice. So you can see here that the differentially expressed genes in the males versus the females are very distinct. There's very little overlap. And we also identify far more genes when we analyze the sexes separately versus the sex collapse data set, which indicates that there is this treatment by sex interaction that you can't detect unless you're looking at an interactive model, but it's informative to do the analysis separately as well. So when we look at our network analyses, this is for our sex collapse data set, you can see a lot of pathways that are involved in development. So we have embryonic organ morphogenesis, regulation of response to food, anterior-posterior pattern specification, and tissue development. And here, these are differentially expressed genes in red. They are upregulated in the morphine-treated pups, and in blue, they're downregulated in the morphine-treated pups. So generally, we're seeing a trend for disrupted processes in neurodevelopment, or development, just generally speaking, which we would expect. Interestingly, when we look at this, when we look at the enrichment of differentially expressed genes in the males versus the females, we identify these clusters of genes. For the females, here we see a cluster of ribosomal genes. So these are RPL, RPS genes, which encode different subunits of ribosomes and that machinery, as well as these NDUF or mitochondrial genes, which play a role in oxidative phosphorylation. And in the morphine-treated females, both clusters of these genes are all upregulated. However, when we look at the males, we see a slightly smaller cluster of RPL, RPS genes and a smaller cluster of mitochondrial genes. However, in our morphine-treated males, these are all downregulated. So we're seeing a really interesting opposing effect on the regulation of genes involved in translation and metabolic function between the males and the females. So this is just a brief summary of what I found are different phenotypic indications of opioid withdrawal, some of our effective findings and our spatial memory deficits, decreased anxiety-like behaviors, as well as an increased locomotor response to methamphetamine during late adolescence. And lastly, that both sexes show disrupted developmental processes, and we see this interesting sex-specific effect on the regulation of ribosomal metabolic / mitochondrial gene networks. So I just want to thank everyone in the lab, Cameron Bryant, my mentor. We are small but mighty. And with that, I will take any questions.
Great talk. I think we have a question from the chat. So they're asking you, have you examined whether particular ranges of USBs that morphine-treated animals express, predict, or are related to behaviors in adolescence? And then there's another one on top also.
Right, so ranges of USVs. So we've done a more in-depth analysis of our USV data. I wasn't responsible for that analysis exactly, but we did look at the different syllables that, so in USVs, you can have different waveforms of the actual sound bites. And we can see that beyond just elevated rates of USVs, that our different treatment groups and different sexes show different profiles of the syllables that they're actually producing in their USVs. I don't know exactly what those findings were, but that should be published somewhat shortly.
Kristen, in your work in the CFWs, it looks like you're up to about 80 individuals in the plots that you showed. Is that also true for the RNA-seq?
No, so the RNA-seq is a much smaller sample size. So all of those results are preliminary. We have a total of 12 mice in that analysis and 6 morphine and 6 saline. So all of the sex-specific findings are just from 3 versus 3 in each treatment group.
Abraham Palmer, and of course, Cameron knows this better than I do, but there's expression data for about 55 CFW animals for hippocampus, striatum, and I think prefrontal cortex. And I'm just wondering what alleles of mu-opioid receptor are segregating in the CFW, whether you can partition your NOWS animals on the basis of mu-opioid receptor status, which is known to contribute to the withdrawal in human infants, at least, to the extent that one can do this kind of study in a human.
Mm-hmm. Yeah, we haven't looked into that, but that's a really interesting idea, and we probably will now.
There are about 2 or 3 human genetic studies that are all the classic association studies, and therefore very fraught in terms of false discovery. But BDNF and mu-opioid receptor and a few others have been supposedly tagged. I don't personally believe it, but in the CFW could certainly address that issue pretty efficiently.
Mm-hmm.
Okay.
Thank you.
I think next, thank you, Kristen. Next, I think we have a coffee break from 1:10 to 1:25.